Hello guys. So as promised, I told you that I will be helping you to be in touch with the topic. This will be an attempt for you to be helpful in reading the textbook. Now, do you remember every time we keep on saying that you should read the textbook, ma'am, but we don't get time to read. Hardly we are able to understand Parsons. You know, Parsons is an important book, but yes, it is much more than the lines itself. So you have to read between the lines in order to get the exact data what they want to give. And it is not just a UG level book. It is actually documented and advised for the people who are going up for your PG residency in ophthalmology. So I thought, why not helping you to read and understand this textbook, and let's take advantage of the extra time that the NMC people have given you. So we will take it as a challenge. We will not waste this time, okay? And we will utilize this time to get maximum extract and maximum benefit. So what I'm going to do, we will be going topic wise topic, and I have named this series as the Parson Series, so that whatever things are important, okay, we'll be dealing them line by line. So this is your first topic, the embryology and the anatomy. And as it goes, in our classes also, whether it is foundation or regular, I usually start the ophthalmology with the anatomy and development only. This is the first, topic of my index, whether it is in classes or whether it is the app. Those who read from me, they know. And we'll try to first of all decode the first very topic from the Parsons. So if you see here, what they are trying to say, the central nervous system is developed from the neural groove, and this neural groove is actually this neural tube is actually going, okay, and it is invaginating. And it is invaginating to form the neural tube. So our development, if you remember, starts from there only. I tell you that we have an optic vesicle, we have got a lens vesicle, then we have got the surrounding mesenchyme, and we have a mesoderm also. So basically they have started with the neural tube. It is running down longitudinally down the dorsal surface of the embryo. Now at either side from the lateral aspect of the anterior portion, which is a precursor of the forebrain, a thickening occurred. So basically they are trying to tell you about the optic vesicle. Basically they are trying to tell about the optic vesicle, and you must have gone through the embryology video also earlier. I have told you all the sequence of events that are taking place in the development of the optic vesicle. First, it was forming a thickening, that is called as a placode, and then it was a small pit kind of a thing, then it was a sulcus, and then it was a vesicle. So basically they are telling you the same. See here, they are telling you that first is the thickening. Thickening appears at an early stage, which then grows outward as a diverticulum. So words are different, but the meaning are same. Thickening is called as a placode, and this diverticulum is first forming a pit, and when it becomes even more deeper, okay, I hope this is visible to you. When it is forming and going even more deeper, then it is forming the sulcus. So it is this way. So it is first, first it is the thickening, then or it is called as a placode, then we have a pit, we have got a sulcus, and finally it is forming the vesicle. So this is how the sequence of events which are taking place in the cases of optic vesicle, and finally it is forming your optic vesicle. And optic vesicle is the structure which is actually representing the what? It is actually representing the neuroectoderm. Now this is again important, that this is representing the neuroectoderm. So whatever are the structures which are developing from this optic vesicle will be representing the structural derivatives of neuroectoderm. So see this. Now from this pair of the diverticulum from the sides and the mesodermal and ectodermal structures are in contact, the two eyes develop. So this was the starting, how actually the first eye-like structure is developing. Now after it meets the surface ectoderm, okay, we have got two parts of the ectoderm. One is your neuroectoderm and one is your surface ectoderm. So when it is meeting the surface ectoderm, this is invaginating from below and it forms a optic cup. So this we already know also, if you remember, how was your this optic vesicle was there, and then it was elongating to form the optic stalk, and this structure was your optic vesicle. If you remember, this was your optic stalk. This optic stalk forms the optic nerve in the future. This forms the optic nerve, and this structure which you are going to see, this is your optic vesicle. This is your optic vesicle, right? Now when it reaches near the surface ectoderm, when it is reaching near the surface ectoderm, something like this, okay, that is why I'm saying that we have to read between the lines, okay. So see, something like this, when it is reaching near the surface ectoderm, okay, then here also it starts showing the invagination and thickening. So we have a thickening in this area, then it also starts showing the invagination. So from here starts the sequence of this lens. First we have the lens placote, that is your lens thickening. Then we have got the lens pit, right? Then we have got the lens sulcus. Usually we do not tell the sulcus in the lens, but it can be there, okay. And then we have the lens vesicle. Then we have the lens vesicle. So if you look here, this was your optic stalk and this was your optic vesicle, and now we are having something like this. This was your surface ectoderm, from which the lens vesicle has come out. So this is your lens vesicle. Okay, now this lens vesicle is causing the changes in this optic vesicle, due to which we start having the depression here. So we will be having a optic cup, something like this, and this will be taking up the lens vesicle here, and here I will be having this surface ectoderm. Here I will be having this surface ectoderm. So this will be your final outcome. Can you see? This is your surface ectoderm. This is your lens vesicle. This is your lens vesicle, right? And this is your optic cup, and this is your optic stalk. This is your optic stalk. So from the neural plate, the neuroectoderm was coming, and how it is causing the changes, we have seen this. Okay, now let's go back. What they are saying, after it meets the surface ectoderm that I have already shown you, and the primary optic vesicle invaginates, forming the optic cup, right? Now the inner layer of the optic cup forms the main structure of the retina, and the nerve fibers from which eventually grows backwards towards the brain. Now what they are telling you, they are telling you the derivatives of this optic stalk, optic cup, lens vesicle and surface ectoderm. So what we had already discussed also, that the outer layer of the cup forms the outer pigmented layer of the retina, as well as the ciliary body, as well as the iris. And if you look at the inner layer is forming the inner layers of the retina, that is your neurosensory retina. It is also forming your secondary vitreous, it is forming tertiary vitreous, it is also forming the muscles of the iris. So see here what they are saying that the outer layer of this cup forms a single layer of pigment epithelium, that you know. Then we have a space between the two. And what was this space? The subretinal space. So between the two lines is a narrow space that is representing the original optic vesicle. Okay, and from its anterior end, so this outer part of the optic curve which is forming posteriorly the pigmented layer of the retinal pigment epithelium, that is retina, when it is going anteriorly it is forming this ciliary body also, outer epithelium, and the iris also. So basically the outer layer is forming the pigmented epithelium of the retina, of the ciliary body, and the iris, right? And at the point where the neuroectoderm meets the surface ectoderm, the surface ectoderm was also showing the thickening. So that also I have shown you, that is in the form of the lens placode, and ultimately it is forming the lens vesicle. So what is the thing that they have told you? They have told you that the if you have this optic cup, okay, optic cup may I am having the outer layer, and then we have the inner layer. Outer layer and inner layer. So they have talked this outer layer is forming the pigment epithelium. It is forming the pigment epithelium of the retina. So that means it is forming the outermost layer of the retina, that is pigment epithelium, RPE, also of the ciliary body and the iris. So basically the epithelium of the cornea, conjunctiva, lacrimal apparatus is forming from the surface ectoderm, but the pigment epithelium of the retina, of the ciliary body and the iris, they are coming from neuroectoderm. While if you look at the inner layer, this is forming the neurosensory retina, that is the inner part of the retina. This is forming the neurosensory retina. Then it is also forming the secondary as well as tertiary vitreous. Always remember the anatomy. In front of the neurosensory retina, what all is lying is the secondary and tertiary vitreous, because tertiary vitreous is nothing but the ciliary zonules, right? And then either again in front of it, what else do we have? We also have the iris muscles. Now this is again important because iris muscles, that is the smooth muscles of the iris, are not coming from the mesoderm. They are actually coming from this neuroectoderm. Okay. Now going back, now the hyaloid artery. If you look at the hyaloid artery, it enters the optic cup through the embryonic fissure and grows forward to meet the lens, bringing the nourishment to the developing structures. Now what happens when the optic cup and optic stalk are growing? It is something like this. This is your optic stalk and this is your optic cup like this. This is the optic cup. Now this area is the optic stalk. This optic stalks forms the optic nerve. That is also very, very clear. Now what was happening? This lens vesicle that was separated from the surface ectoderm. So this optic cup starts, invaginating around this lens vesicle. So in doing so, how it is growing? It is growing superiorly and laterally. Again, this I told you in the development of the eye also. So it is growing something like this. It is growing superiorly and it is growing laterally. So there was a deficiency that was created, embryonic fissure kind of a thing is formed, which is also called as a choroidal fissure. And that is why when we have got the colobomas, they are typically present in the inferonasal quadrant, because originally also we have a deficiency inferiorly and nasally because it is growing superiorly and laterally. Now, when we have the surrounding mesenchyme, which is surrounding the neural tube that is growing, so from that what happens, the vascular part, it is actually passing through this area. It is passing through this optic stalk and it is going to the retina. So this remains as the hyaloid artery. So what happens after its entry into this optic stalk, it gets obliterated here. So that is why this hyaloid system of the vessels becomes completely cut off from its original vascular layer. Okay, now see, the same thing they have said, that the hyaloid artery enters the optic cup through the embryonic fissure, grows forward to meet the lens and bringing the temporary nourishment to the developing structures before it eventually atrophies and disappears. And its place is then taken up by what? Afterwards, its place is taken up by the vitreous, and this vitreous is also largely secreted by neuroectoderm. So you know that in front of the retina, suppose we are talking about the retina, something like this, it is there. This is your iris, and we have the ciliary body. Okay, so this retina, and in front of this retina we have got what you call as the vitreous gel like structure, something here. So this vitreous gel like structure, this is also forming by the neuroectoderm. Then the ciliary zonules, they are also forming by this neuroectoderm. If you look at the ciliary zonules, so here we have the lens and these zonules, they are also part from this neuroectoderm. So basically this structure is showing that all these structures which are present posteriorly, like starting from the RPE, then the neurosensory retina, then secondary vitreous, the tertiary vitreous, the ciliary zonules, okay, all these things are actually the derivatives of neuroectoderm, while the hyaloid artery, that is a derivative of the mesenchyme, that is representing the neural crest. And after some time, when it is passing through this optic up, the the origin gets obliterated, and now it is really cut off from the surrounding places, right? Now after this, they are saying that while these ectodermal events are taking place, what else is happening? So while these ectodermal events are taking place, at the same time, the mesoderm is also differentiating to form the coats of the eye as well as the orbital structures. So meanwhile, we have got other things also, and these are forming from the mesoderm. Now that are present between the lens and the surface ectoderm becomes hollow to form the anterior chamber lined by the mesodermal condensations, which forms the anterior layer of the iris. So meanwhile, we also have the mesoderm. Now what happens with the mesoderm? If you look at the mesoderm, it is condensing and it is actually forming both the parts, the anterior part as well as the posterior part. So what happens, anterior part will be forming the anterior part of our eyeball, while the posterior part will be forming the posterior part of the eyeball. Like for example, if you see here what they are saying, the anterior part. Now if I am talking about the iris per se, so that is forming the anterior layers of the iris. Then you have got other structures like uske piche I have got the angle of anterior chamber, we have got the main structures in the cornea also. While if you look at the surface ectoderm, so I told you that the surface ectoderm is actually responsible for the epithelium. What was the mnemonic for surface ectoderm? Do you remember that? For the surface ectoderm, we were having the LECs. This is the mnemonic. That means L was lens here, then E. E was the epithelium. Epithelium of the conjunctiva as well as the cornea. So we have got 2C here, and also epithelium of 1L. So this is epithelium of the lacrimal apparatus. Epithelium also of the lacrimal apparatus, and along with this we also have lacrimal gland. So how many L we have? We have got L3EC square. So lens epithelial cells, all these structures are forming from the surface ectoderm, while iris muscles are coming from neuroectoderm. Okay. Now come back here. So they are saying that it is the surface ectoderm which remains as the corneal as well as conjunctival epithelium. Now if you see, they have given you the summary of the ocular embryogenesis in the table. We'll try to see that table also. And they are saying that ectoderm is of two types. One is the neuroectoderm, one is surface ectoderm. Neuroectoderm is coming from the neural tube that we have already discussed, while the surface ectoderm is coming on the side of the head. Now in the table 1.1, they have given you whole of the summary of the ocular embryogenesis. Many times I get this message also from the students that do we require to learn this? So actually you can be knowing the sequence of events, but it is not particularly required to learn every week. Like, okay, we know mota that it is starting at third week of period of gestation and it is going till 9 to 10 weeks. So you should know this, that first it is the thickening that is taking place. then we have got the pit, then we have got the sulcus, then we have got the vesicle. But I don't think that you need to learn that exactly at 3 weeks we have the optic groove, and then at 4 weeks at the optic plate, something like this. And moreover, you know, when you go to the different books, exactly they will not be same at every place, because you know, medicine is like this only. You won't get embryogenesis exactly at one day in all the people. So it is rough. So I don't think that it is really required. Rest is your wish, okay? Then see here. Now we are starting with the anatomy. Now if we talk about the globe per se, so the wall of the globe is actually composed of the elastic tissue, and it is containing two important things. One is your transparent cornea and the opaque sclera. So wall means that we are talking about the fibrous coat. Okay, so fibrous coat is actually consisting of two things. One is your cornea and another is the sclera, and we all know that cornea is transparent, sclera is opaque. Both are the parts of the fibrous tissue, but both are different. Now the anterior part of the sclera is covered by a mucous membrane. Then if I talk about just the anterior part of the sclera, then it is covered by a membrane. So that membrane is actually the translucent membrane. That membrane is a translucent membrane, and that is nothing but the conjunctiva. So what do you mean by translucent? Translucent means which is something between the transparent and opaque, which has the properties lying somewhere in between of transparent and opaque. You know, cornea is transparent, sclera is opaque. So in between that, through which the things are semi or partially visible, that is your conjunctiva. And it is actually something which is required to connect the eyelids to the globe, because otherwise what will happen? I have got these eyelids and I have a globe, and I have nothing which is connecting the two. So I should have something which is connecting the two also. So for that I have the conjunctiva, which is reflected from its surface onto the eyelids. So when you are reflecting this, suppose it is something like this. So when you are reflecting it, what we are getting is the conjunctiva, right? Now inside the eye, posteriorly the sclera is lined by another tract. So we have already talked about the fibrous coat here. Now after that they are saying that there is one more coat, and that coat is actually the uveal tract. Okay, so this is your uveal tract, and then the third thing is the retina, and they are broadly divided into the anterior segment and the posterior segment. So again, what we talk about this in the classes also, that eye is actually the eyeball. Okay, and this eyeball has got three coats. They are talking about this, that eyeball actually has got three coats. We have already talked about one, that was your fibrous coat. Then the second is your vascular coat, and the third is the nervous coat. Okay, now in the fibrous coat, you already know we have got the cornea and the sclera. Vascular coat is your uveal tract, right? And uveal tract consists of the iris, the ciliary body and the choroid, while the nervous coat consists of the retina and the optic nerve. Retina as well as the optic nerve. So these are the various coats of the eyeball. Coats we can say, or the layers or the tunics of the eyeball we have. And we also know that the inside of the eyeball can be divided into two segments. So if this is the eyeball, suppose, okay, now what is happening? This is the iris here, and we have this ciliary body, and then we have the choroid, which I have drawn with the red color because it has the choriocapillaries, and then we have the lens here. It is holded by the ciliary zonules here, and then we have got the innermost structure also, that is your retina. Now what they are saying that there is another term that is used here, and that is actually the segments. So these segments are actually two. We have got two segments in the eyeball, and whenever we have got two things, it is usually like one is anterior and one is the posterior. One is anterior and one is posterior. So basically, you know, this is the iris, and this iris is actually responsible for dividing the anterior segment itself. While if I talk about the second thing, and that is the lens. Lens is responsible for dividing the eyeball into the segments, and iris is dividing the segment into the chambers. So what happens if you divide this at the level of the lens? So anterior to this, I have anterior segment. Anterior to this, we have anterior segment. While if I look at the posterior side, so posterior to this, we have posterior segment. Posterior segment. While this anterior segment can be divided with the help of the iris. So this structure is your iris. So this iris is further dividing this anterior segment into two chambers. So I have this anterior chamber and then this posterior chamber also. So whenever I am talking, you know, in terms of academically also, like some are specialists of anterior segment, some are specialists of posterior segment. I can talk clinically also. When I'm talking about the pathologies, that this is the pathology of anterior segment, this is the pathology of posterior segment. So like way we are always using the segments as well as chambers. So see this, they are saying that it is the iris which is dividing the anterior segment. Anterior segment. The iris is dividing this anterior segment into anterior chamber bounded by the cornea anteriorly and posterior chamber bounded by the lens posteriorly. Now you know that the anterior segment which contains both the chambers. Anterior segment which contains both the chambers, that is your anterior chamber as well as posterior chamber, they contains the same fluid. This fluid is actually the aqueous humor. This fluid is aqueous humor and its consistency is just water-like. That is why it is called as aqueous humor. The refractive index is 1.33 and it contains 99.99% of water. Now, because this contains 99.99% water, that is why it is called as aqueous humor. So, see, they are saying that this cavity is actually filled with a clear watery fluid called as the aqueous humor, while the posterior segment is something which is present behind the lens. So, that cavity which is being bounded, that is called as the vitreous cavity. So, this posterior chamber in the posterior segment, okay, it is the segment which is lying behind the lens. Now here actually they have used one word incorrectly because this will give you a false impression. They are saying that in the posterior segment, the posterior chamber lies behind the lens. So this line is actually incorrect. They have used a wrong word here. Actually it should be like the vitreous cavity. It is the vitreous cavity which is lying behind the lens and not the posterior chamber. Posterior chamber is lying behind the iris and in front of the lens itself. And this contains another, you know, substance which is having gel-like consistency, transparent gel-like consistency, which is called as vitreous humor. So in this section, we have seen that how the development of eye takes place, what is responsible for maintaining the shape of the eyeball, what are the different parts of the eyeball, and what are the different segments, how they are divided into anterior chamber and posterior chamber, and also what are the different fluids present in the eyeball. So that was all for today's session. I don't want to make it like pushing your limits. I want to keep it crisp and short, so that daily you can give 15 to 30 minutes for your textbook reading, which is like a golden pearl. You can never, replace your textbook with any of the things. So it's always good that you are able to read your textbooks. And in your busy times, in your COVID times, in your so many difficulties, when you were not able to read the textbook and having so many difficulties, I try to help you. Like, you have to do the things, but there is someone who is there to help you and always there to support you, and that is me with my ophthalmology, so that you don't have to work and struggle with the book, and in no time we will be completing all of the series. So just give 15 to 30 minutes daily, and we are going to deal with everything in the Parsons. Then some of the tables, some of the flow charts, some of the diagrams which I feel that are important, we will also be marking them, so that we can save those flowcharts, those pictures in our smartphones in a separate folder that will be called as the Parson folder under the Parson series. And just before giving the exams, we can have a quick look and you are through. So hope you like this session. Tell me in the comment section what you feel about this, and how you are going to be benefited from this, how often you want this Parson sessions to be done. What is the idea if I give you daily 15 minutes to 30 minutes, or you want it at more of the distances. I like, from my side, I want you to give a daily or alternate, so that in this period of time, like your NEET exam has been postponed, we can comfortably get finished up. This will not overburden you. You can carry on with your timetable, and also you can complete your textbook reading. Thank you, and happy ophthalmology.